Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing GATE Biotechnology question paper conducted in the year 2023. I will be discussing the general biology and rDNA. If you want 2024 GATE Biotechnology question papers answered, you can go to my channel Omni Bio Entrance, and in the video section you will find. Then next question. The event that lead to the inactivation of tumor suppressor genes in cancer cells is or are. So, what are these tumor suppressor genes? They prevent cancer. They regulate cell growth and thereby prevent cancer. Now, in cancer cells, the tumor suppressor genes which are present in normal cells gets inactivated. Now, how is this tumor suppressor genes getting inactivated? Is it gene amplification? No. Gene amplification, it lead to oncogene activation or cancer cell activation. Promoter methylation, yes. If you add methyl groups to the promoter region of a tumor suppressor gene, it can silence the expression of the tumor suppressor gene. Then loss of heterozygosity, that is also correct. Now you receive one gene from your parent and the other from male parent and the other from the female parent. Now suppose if the gene that you received from the father is mutated or deleted, and the gene that you received from the mother or the allele of this gene that you received from the mother is also non-functional, then the tumor suppressing function will be lost. So if there is loss of that is loss of heterozygosity. So if there is loss of heterozygosity, then also can lead to inactivation of tumor suppressor genes in cancer cells. So B and C are the answer. Then next one. Methylation of CpG islands near the promoter of a gene can inhibit transcription by preventing RNA polymerase binding. That's correct. Facilitating repressor binding. That is also correct. Facilitating heterochromatin formation. That's also correct. It can lead to more compact and condensed chromosome, that is heterochromatin formation. So that because of this heterochromatin formation, it will be inaccessible to transcription factors and RNA polymerase. So A, B, C is the answer. Inducing euchromatin formation. It is not euchromatin, it is heterochromatin. Then next one. Which of the following statements are true about induced pluripotent stem cells? Now what are these induced pluripotent stem cells? So induced pluripotent stem cells, they are created or generated by reprogramming the somatic cells to come back to the pluripotent state, that which is similar to your embryonic stem cells. So, which of the following statements is or are true about induced pluripotent stem cells? So, they can self-renew, yes. They can proliferate and self-renew in vitro conditions. So, that's correct. They require specific signal to maintain their stemness. Yes, to maintain their pluripotent stem nature, they require specific signal, like they require certain transcription factors, growth factors, etc. to maintain their pluripotency. So B is also correct. They cannot be genetically manipulated. They can be genetically manipulated using CRISPR-Cas9 editing tools. Then they can form organoids in vitro. Yes, that is also correct. Because they can differentiate into various cell types and form organoids in vitro. So A, B, D is the answer. Then next one. Which of the following statement is or are true about fluoroquinolone drugs? So they contain quinolone rings, that is correct. They inhibit RNA polymerase, not RNA polymerase. They inhibit bacterial topoisomerase or the gyrase. So A and C are the answer. Then next one. Which of the following is or are plant protoplast fusogenic agent? Sodium nitrate is, polyvinyl alcohol, yes. Polyethylene glycol, yes. Then bromoxynil, it is a herbicide, not a plant protoplast fusogenic agent. So A, B, C is the answer. Next one, direct DNA transfer method used for plant genetic engineering. So in direct DNA transfer method, it involves directly injecting or directly transferring or directly transforming the plant cell with DNA, and it doesn't involve the use of any vectors like agrobacterium tumefaciens. So microparticle bombardment, yes, it is a direct DNA transfer method, and it uses gold or tungsten particle which are coated with DNA and uses high pressure to transform plant cells. Then electroporation, it uses electrical pulses to create temporary pores on the plant cell membrane, allowing DNA to enter. Then polyethylene glycol, it is used to facilitate the uptake of DNA by plant protoplast. So B is also correct, C is also correct. Agrobacterium mediated transformation is not correct. It is not a direct DNA transfer method, it is indirect DNA transfer method. Then next one, which of the following vectors is or are used to clone a DNA fragment of size 220 kb? It is bacterial artificial chromosome can be used and yeast artificial chromosome can be used. Whereas cosmids, they can accommodate up to 40 to 50 kb and PUC19 can go up till 10 to 15 kb. So A and B is the answer. Then next one. The following equation reaction represent biomass synthesis from hexadecane. So this is hexadecane, where CH 1.66, O 0.27, N 0.27 represent the biomass. So this is the biomass that is produced from hexadecane. It has used 12.5 moles of oxygen and has released 5.37 moles of carbon dioxide, and they have asked what is the respiratory quotient. So respiratory quotient, it is given by the formula moles of carbon dioxide produced by moles of oxygen consumed. So substitute the values and you will get 0.43. Then next one, temperature of a reaction with an activation energy value of 15 kilocalories per mole is increased from 300 to 310 Kelvin. If the value of the ideal gas constant is this, the ratio of the reaction constants K310 by 300 is. So you know the formula, that is ln K1 by K2 is equal to EA by R into 1 by T1 minus 1 by T2. EA is activation energy, R is gas constant, T1 is 310 and T2 is 300. Then you will get ln K1 by K2 will be 0.811 after substitution of the values. K1 by K2 will be 0.811 divided by 2.303. So that will give you 0.3524. 2.303 is for conversion of ln to log value. So then you have to find the antilog of 0.3524, that is 2.25. So the answer here is 2.25. Then next one, E. coli cultivated at 298 Kelvin uptakes an uncharged compound by passive diffusion. The intracellular and extracellular concentration of the compound A are 0.001 molar, that is intracellular concentration, and extracellular concentration is 0.1 molar. Ideal gas constant is 1.9872 calories per mole per Kelvin. the free energy change in kilocalories for passive diffusion. So the delta G for passive diffusion is given by minus RT ln C out. That is C out is 0.1 and C in is intracellular, so that is 0.001. Substitute the values, you will get minus 2727.115 calories per mole. They have asked in kilocalories per mole, so convert it, you will get minus 2.7 kilocalories per mole. Then next one. If there are 3 unrooted trees for 4 protein sequences, the number of rooted trees for the same number of sequences. So here the number of rooted trees is given by this formula. That is 2n minus 3 factorial by 2 raised to n minus 2 into n minus 2 factorial, where n is the number of protein sequences. That is, here it is 4. So substitute the values and you will get 15. Then next one. An enzyme E catalyzes the biochemical reaction A to B with Kcat equal to 500 per second if the initial velocity is 10 and the total enzyme concentration is 30, substrate concentration 40, the value of Km in micromolar. So they have given Kcat and enzyme concentration which is in nanomolar. So convert it into micromolar. Okay. So Kcat is given by the formula Vmax by Et. So from there you can calculate Vmax. So Vmax is Kcat into enzyme total. That will be 15 micromolar. Now use the Michaelis-Menten equation. That is V0 is equal to Vmax concentration of S by Km plus concentration of S. From there you can calculate Km. Substitute the values. Vmax we have found out. S they have given as 40 micromolar. And V0 is 10 micromolar per second. So substitute the values and you will get Km value as 20. Then next one. DNA sample collected from an unidentified bacterial species contain 13% of adenine. The GC content of Y is. Now 13% of adenine means then a number of moles of or number of or percentage of thymine is also 13. So it will be both combined together will be 26%. Now 100 minus 26 will give you 74%, which is G and C combined together. So it is 74%. That is what they have asked. That is GC content in Y. So it is 74%. Then next one. If 1000 base pair of a double helical DNA weighs 1 in 10 raised to minus 18 grams and the distance between two base pair is 0.34 nanometers, the total amount of DNA required to stretch from Earth to Moon is how much? And they have given distance between Earth and Moon. So distance between Earth and Moon, that you have to convert it into meters. So that is 3.74 into 10 raised to 8 meters. Now distance between two bases is 0.34 nanometers. So convert it into meters. So it will be 0.34 into 10 raised to minus 9 meters. Now you calculate the number of base pairs required. That is total distance by distance between two base pairs. You will get it as 1.1 into 10 raised to 18 base pairs. Now weight of 1000 base pair DNA they have given as 1 in 10 raised to minus 18 grams. They have asked for total amount of DNA in milligrams. So weight of this much amount, this much base pair of DNA will be, substitute the values, divide by 1000, you will get it in grams. Convert grams into milligrams, you will get it as 1.1 milligram. Then next one. A DNTP master mix is prepared by combining 40 microliter each 20 millimolar DNTP stock, that is ATP, CTP, GTP, TTP, Then from this master mix 4 microliter is taken and added to PCR mix. And the final volume is made up to 50 microliter. Now the concentration of total DNTPs in the PCR mix you have to calculate. So 40 microliter of each 20 millimolar DNTP stock is combined. So the total volume of the DNTP master mix is, so 40 microliter, 4 DNTPs. So how much it will be? 40 into 4, that is 160 microliter is the total volume of the DNTP master mix. Okay. Now the concentration of each DNTP in the master mix. So 20 into 4 divided by 160, that is by using M1V1 is equal to M2V2 formula. So M1 will be 20 millimolar, V1 will be 40, and M2 we have to find out, and 160 is V2. So you will get 5 millimolar. Now there are 4 DNTPs. So the total DNTP concentration in the DNTP master mix will be 5 into 4, that is 20 millimolar. That is in the master mix. Now from this 20 millimolar stock you are taking 4 microliter and is added to the PCR mix and made up the final volume to 50 microliter. Then they have asked the concentration of the total DNTPs in the PCR mix. Again M1V1 is equal to M2V2. M1 will be 20 millimolar. From there you are taking 4 microliter. So 4 is equal to how much? M2 into final is made up to the PCR mix is made up to 50 microliter. So into 50. So M2 will be 20 into 4 divided by 50, that is 1.6 millimolar. Now convert to micromolar, you will get it as 1600 micromolar. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.